Hello, everyone, and welcome to today's webinar. I'm Iris Kobatsky, Assistant Science Editor for The Scientist, and I will be moderating our discussion. Dr. Svea Cheeseman will be taking his place and will present data on two studies where the automated mass photometer 2 Auto was used to evaluate the oligomeric status of proteins when subjected to various experimental conditions. Svea Cheeseman is the Director of Product Management for Cell and Gene Therapy at Refeyn, responsible for products targeted at the biopharma industry. She holds a PhD in single molecule biophysics and has 5 years' experience in the biopharma industry. She has a keen interest in pushing the boundaries of biomolecular analysis to eventually enable real-time release of biomolecular drugs. Okay, let's make sure our slides are up and running. Perfect. Dr. Cheeseman, the floor is yours.
Thank you very much, Iris, for the kind introduction, and welcome everyone to this seminar. At Refeyn, our mission is to transform analytical workflows in the life sciences and the biopharma industry using the power of light. And we want to answer key questions in biomolecular research and development, such as questions around sample characterization. Is the sample pure enough for the next stage of analysis? Are the gene delivery vehicles filled, such as AAVs? Or what conditions optimize protein purification? Also, we want to address interaction studies with questions such as what is the complex stoichiometry, how does the complex form, and how strong are the interactions? And finally, we also want to analyze, to help with the analysis of complexes with questions like what is the composition of the functional species, how many different complexes can form, or what are the conditions that stabilize these complexes. We think that mass spectrometry is the right technology to address those questions because it determines the mass in bulk with single molecule resolution. It also has a wide measurement and application range. It is easy to use, is quick, and consumes very low samples. And it also has very minimal sample preparation as it is label-free and immobilization-free. And finally, in this seminar, I would also like to speak about the automation of mass spectrometry, which combines the sensitivity and simplicity of mass spectrometry with the ease and efficiency of automation. But to get started, I'd just like to introduce mass spectrometry a little more. So we can measure the mass of molecules in the range of 30 kilodalton to 5 megadalton, and we're not limited to any specific molecule. So in the low range, we could, for example, we can see proteins such as monoclonal antibodies, but also smaller ones. We can see protein complexes and all the way up to macromolecular assemblies. But also nanostructures in the megadalton range. Nanostructures in small viruses such as AAVs can be measured with mass spectrometry, and also nucleic acids. In the low range, this could be DNA fragments or RNA fragments, both single or double-stranded. And also plasmids. I'll show examples of most of these classes during this presentation. And how do we do that? So basically, mass spectrometry weighs molecules with light. And in this cartoon, it's quite nicely shown how we do that. Basically, a small molecule is a light molecule and sheds, if you shine light on it, it sheds a very small, gives a small shadow. And the bigger the protein, the bigger the shadow, the darker it is. This is sort of what we do. So in more detail, we measure on the glass surface. You add your molecule in solution onto the glass. And naturally, the molecules would approach the glass surface. When they do, that's when we measure them. And what we actually measure, we shine a laser on them. And we see the interference of two phenomena. One is the reflected laser light from the glass surface, which is always the same. And the second one is the scattered light from the biomolecules that sit on the surface. Now, scattering is interesting because it scales with the optical density and the size of the scattering object. And that means that we can use that to determine the mass of it. So what we see when single molecules come to the surface, we see on the camera chip this interference pattern. This is the interference of the scattered and the reflected light. Now, the more scattering there is, the darker the center is, the contrast. So that is what we measure, the contrast on the chip. And we can then calibrate the system with a protein of a known mass. And the contrast linearly scales with mass. So when we do that, when we calibrate the system, you can see that the expected mass of different sized molecules, proteins in this case, scales linearly with the measured mass on the mass photometer over the entire range. Now, the output of mass photometry is a histogram. So initially, it is counts over the contrast. And once you calibrate the system, it is counts over the molecular mass in kilodalton. And each of these events is a single molecule. So we do have single molecule resolution. Now, before starting to talk about more about the automation, I'd like to bring us all to the same page in terms of what the applications of mass photometry are and where the benefits lie. So let's start with the sample characterization. A very popular application for mass photometry, especially used by structural biologists, is to quickly check whether the sample is what you expect it to be. So this is an example where people looked at the HTT, which is a protein altered in Huntington's disease. And before they proceeded with further analysis, they just wanted to quickly check whether the sample binds to the HAP40 as expected, and whether the sample is clean. And so they could quickly do that with a mass spectrometer. Just it takes less than 5 minutes and 20 microliters of sample. And they could see that, yes, here, this dominant peak, this is the HTT with the HAP40. There is a dimer of that complex too, but very small percentage, and a few proteins at lower range. But this was, for them, the confirmation that they can go on with their application. So this is really useful to do when, especially when the following analysis is very time and cost consuming, such as electron microscopy. Mass photometry can also be used to optimize processes. So what we look at here is the 700 kilodalton 20S proteasome of rats. And we look at the different purification steps. So basically, a sample was taken after all of these purification steps from early elution gel filtration, late elution gel filtration, second chromatography, buffer exchange, and the final column. And the authors looked at this peak. This is where they expected the 20S proteasome to sit. So we can see that the further the purification progresses, this peak increases relative to the other molecules detected. However, now they also plotted the same thing. They plotted here the percentage of this peak over all molecules detected. And they could find that using their established process, the last step actually leads lower purity of the 20S proteasome than the fourth step. So basically, they learned during this analysis that the fifth step can be omitted. Mass spectrometry is not limited to proteins. What you see here is a polymeric micelle. So in this case, they use polymeric micelles to encapsulate a water non-soluble drug. And what they tested here is, on the top is just the empty micelles. So that is just the formation of these micelles without the drug. But with different amounts of dendrimers. And we can see that if you have 4 dendrimers, we have a much lower weight than if you have 8 dendrimers. So mass spectrometry can be used to see whether the expected size is reached and its expected mass, but also it tells you something about the monodispersity and the cleanliness of the sample, right? So here we don't see any further peaks, we don't see contaminations, so nicely assembled micelles. They then loaded the micelles with fulvestrant, which is the drug, at different ratios. So 10% weight ratio and 20% weight ratio on the FLV8 up here. And we now see that the mass peak is shifting towards higher weights. And it shifts more the more fulvestrant is inside the solution. So this shows that the encapsulation is successful. Another popular application of mass spectrometry is the measurement of viruses, specifically AAVs. In the production of AAVs, you want to have the DNA inside the viral capsid, but during the production, you also produce empty capsids that don't have the DNA. Now, the difficulty in detecting this is that they both have the exact same size, right? So anything that looks at size wouldn't find the differences between empty and full. That's why you typically need to separate them beforehand. Mass spectrometry, however, measures mass. And the mass between empty and full is very different because the full ones contain the DNA genome. And it's about a megadalton heavier. So here you can see that we started off with only empty AAVs. This is this peak. And now in these experiments, we increased the ratio of loaded AAVs. And you can see that this is the loaded peak here. And we see that increases the higher this percentage. And we see this really nice linear relationship for the entire range from 0 to almost 100% AAV loading. Just as a side note, this was measured on a dedicated instrument for AAV characterization, the SamuxMP. We can also look at nucleic acids, as mentioned initially. What we've done here is the system was calibrated using a DNA ladder from 100 base pairs to 2,000 base pairs, and then used to measure a PCR product of 331 base pairs, which comes out nicely, as you can see here, and also the PAC18 plasmid of about 2,700 base pairs. So what I show you here means that mass spectrometry can be used to quickly check after the PCR whether your product is there, whether it's pure, or whether your plasmid is intact. Now moving on to another application field, which is interaction studies, the binding of proteins, starting with the workhorse of biopharma, the monoclonal antibody. So here, what we see in the top panel is the monoclonal antibody Herceptin, measured separately from its antigen HER2. So we can see that both when in separation, when just the antigen or just the monoclonal antibody, they lead to these nice peaks, and then they mixed both the antibody and the antigen in a ratio of 1:1. And what we can see now is that one of these antigens is binding to the antibody. This is the blue peak here at the higher molecular weight. The weight is exactly the sum of these two. However, if you increase the concentration to 2 times as many antigens as antibodies, we see the binding of a second antigen to the same antibody, which is shown here. And so I think that everyone who has worked with antibodies before and proteins that bind 2 different partners, they can appreciate that resolving this is often a pain. But because the mass is so nicely resolved and we look at single molecules, mass spectrometry can be used to look at proteins that bind to more than one binding partner. A bit more complex, but a similar study is performed here. So not only did these authors look at the binding of antibodies to antigens, they also looked at the changing conditions. So on the top again is the antibody and the antigen in isolation, but at different pHs. So for the antibody, pH 5, pH 5.5, that doesn't make a difference. The peak is the same. You see a bit more of the low molecular range here at pH 5.5. But for the, sorry, this is IgG, by the way. For the antigen, FcRn, however, there's quite a difference. At the pH 5, you see both the monomer and the dimer, whereas at pH 5.5, at higher pH, it exists basically only in its monomeric form. And at the higher range, there is nothing. So when these two are now mixed, the authors looked at different, the high range, where you expect the peaks corresponding to the binding of antigen and antibody at different pH values. And what they saw, interestingly, is that at pH 5, where, just remember here, we only have monomers, basically. Sorry, at pH 5, we have monomers and dimers. We see single and double binding to the antibody. However, at pH 5.5, where we do not have dimers, in solution, we see a higher percentage, much higher percentage, to be bound by where two antigens bind to the same antibody. This shows us that the very likely pathway of binding of FcRn to IgG is that one binds first and then the second one binds. So in this case, using the screening, we can also determine how the complex is formed. We can also look at binding even though we cannot resolve certain binding partners. So what we see here is a study from AstraZeneca on PROTACs. So in the first window, we have the VHL is the ligase, BD1, BD2 is the target protein with two binding sites. That's the protein that is supposed to be degraded in this process. And then increasing amounts of MZ1. MZ1 is the PROTAC. So the idea of that is that the PROTAC binds to the ligase, brings it close to the, and binds to the protein 2, brings it close, and this leads to the destruction of the protein. So even though the MZ1, so sorry, I should say, we only see one peak here because both of them have the same size, the same mass. Now, when MZ1 was added at increasing concentration, we see the binding of it because we see the ternary complex, the VHL plus the one of plus the protein. Increasing the concentration of the PROTAC further, actually, you can also see the quinary complex here, which is the fully assembled complex. That's the one that you want. Interestingly, now, if you increase further the concentration of the PROTAC, you see the saturation of the binding sites and the inhibition of the complex formation. Finally, I'd like to speak about a few examples for the analysis of complexes. So a very popular one is to look at the oligomerization of a protein. So this is a monoclonal antibody, and it's when you put it on the mass photometer, you can see that it exists predominantly as a monomer, but also as a dimer. You see a little population here as a trimer. And interestingly, you have to believe me that there's exactly one tetramer that was found. Now, this shows that you can very quickly assess the oligomeric state of your protein, but also it underlines that whereas you here in this peak, you see about 3,000 molecules, this is one. So it also makes you appreciate the dynamic range of the mass photometry that can detect species, even if they are 1,000 times less abundant than others in the same solution. This is a study that looked at the formation of the ribosome at the 50S and 30S subunit of E. coli. And they screened different buffers to see in which buffer the protein would form and is stable. So if they used a buffer, like they did on the top, without magnesium, you can see that you can identify the 30S and the 50S subunit, but you don't see anything at the combined weight of the two. However, if magnesium is added to the system, we now see a third peak appearing and even being dominant. That is the formation of the ribosome. Now, what we've learned about the application of mass photometry, we have hopefully learned that it requires low sample volume. It's a fast measurement, takes about 5 minutes. It has a really wide measurement range, therefore application range. It consumes minimal sample and also is quickly prepped and easy to use. Now, we decided that to add a pipetting robot to automate these measurements, which then means that you have an automated operation, you can run multiple samples at once, and you have increased repeatability and consistency. Now, this is what the system looks like. So out here is the pipetting robot, and we have a close-up of the system when the lid is open. So what you see in here is the mass photometer. The mass photometer sits down here together with this anti-vibration solution. It is actually retrofittable. So you can buy the entire system with the 2MP, our current mass photometer. But if you have already a 2MP or even the older version, the first generation 1MP, the system can be retrofitted onto your mass photometer. Now here you see the pipetting robot, fresh pipette tips, a waste, and a 96 wall plate. This is where you place your samples. You program the robot, and here you place a fresh glass slide with the well, and it has 14 wells. So you can measure 14 samples autonomously, which equates approximately to 1 hour of operator-free time. After that, you have to exchange the glass slide and clean the system, put a fresh glass slide on, and you can measure another 14. The benefits of that is that you don't have to do the measurements yourself, but also because a robot is pipetting, you have increased data reproducibility. And that's what I would like to show you a bit further. So here we measured IgG. You see this is the 150 kilodalton, the typical monoclonal antibody weight. We see a monomer and a dimer. And what you see here is the overlay of 10 different experiments that were run in one go on the robot. And you can appreciate that they are very much overlapping. A further analysis down here shows the mass is very consistent of the main peak here across the experiment. While the counts are varying a little bit, I'd like you to have a look at this graph down here, which shows that the result is not affected by measurements in the count rate. Because what is shown here is the percentage of dimers relative to monomers. And this is very consistent across all measurements. So is the mass, despite very small variations in the count rate. So yes, the robot definitely provides higher consistency than a manual operator. Now, what can you do with this? Here's an example where we screened 4 different conditions with statistical significance using triplicates autonomously in the robot. So starting off with an IgG and protein A mixture, a 1-to-1 mixture, at a total concentration of 20 nanomolar of the IgG. And what we can see is that several peaks here are showing up that correspond to either the protein A or just the antibody, and then several combinations of the two of them. So higher order structures. Now sequentially, the total concentration was reduced. And what we can appreciate, the lower the concentration, the lower the abundance of these higher order structures. And where we only show one result here, but these were performed in triplets. So we had 4 different conditions measured in triplets, makes 12 measurements. We have 14 samples on the slide, so we still had 2 slots to measure at the calibrant. We recommend to do the first and the last measurement of a calibrant. This way you have an internal quality control of your measurements in between. What this robot also allows is the automated titration. So what we show here is that again the IgG molecule, and then we added increasing concentration of BSA, the small molecule here. There's 2 things that I'd like you to appreciate here. One is that the counts scale linearly with the BSA concentration added. And something else to appreciate here is that the IgG peak here is very stable. It's not impacted by the increasing amount of BSA. So that shows the robustness of the system, because this small peak could, for example, also be a contaminant in your protein concentrate, in your protein mixture. And despite having a huge abundance of small proteins there, the IgG is quite well resolved and also has the same peak height. So it shows that we're pretty robust against any contaminations. You would see them, but it doesn't affect the measurement. Another example of a titration experiment. This was run by one of the beta testers of the instrument, the Max Planck Institute in Marburg, more specifically the group of Georg Hochberg. And what they looked at is a protein that remains undisclosed, which forms a tetramer when it's catalytically active. That is this peak here. And it forms a octamer when it's inactive. And they have a ligand that if they add the ligand, it promotes the formation of the inactive octamer. This was shown in this experiment too. That this is one run of the robot, where first they started off without any ligand, and we see that most of the population is active as a tetramer. And now with increasing ligand concentration, the concentration is down here, we can see that the population shifts towards the inactive octamer. Now this can also be plotted here to show the dynamics of it, where we see that initially there's a steep increase in octamer formation, which then plateaus at higher concentration. You can also see that each data point has, each concentration has 2 data points. So because they have 5 different conditions, they could run each twice, meaning they ended up with 10 measurements and still had 2, 4 wells free for calibration. You can also use the system for screening conditions. This is another result from Gareth Rogers' group, where they looked at a citrate synthase, which forms catalytically inactive filaments at certain pH values. So they wanted to see what the extent of these is at certain pH values. So they tested the same protein at the same concentration at different pH values, ranging from 6.5 to 8.5. You can see that at the lowest pH 6.5, also shown separately here, you have a very small amount of these filaments. So most of it is active at the peak at 100 kilodalton. And with increasing pH, also the populations of the filaments increased, showing a further inactivation of the protein. Now the system is really easy to use. This is the user interface that you see when you start an acquisition. So here you see the 96 well plate, and it's color-coded. You can define what your buffer is and which ones your samples are. And then you, on the left here, you define the protocol. So you tell the robot what to do, what buffer to use, what sample to use, and also importantly, what dilution. So the system is able to dilute before the measurement. And this is really useful for dilution-sensitive samples, but also if you think about the adsorption to the well plate. So you only dilute it right before you put it on the mass photometer, and this way you reduce the adsorption. This is then the analysis software interface. We have a batch processing functionality. That means that you don't have to analyze every single measurement one by one, but you can load them all into the system and it analyzes them all together. And what our customers really appreciate is this figure design tool that is part of the analysis software, where you can load all of your results into one graph, and then you can design all things about it, you know, what sort of axes you want, what colors, how many measurements are shown, all of that. You can directly export that for your report. We also provide consumables together with the system. So that would be the glass slide and these the silicon sample well cassette. That means that you have hassle-free measurements, you have reduced sample preparation time and greater data confidence. Now, when it comes to the machines available with the automation, as mentioned, our current second generation mass photometer, the 2MP, is available inside the robot. If you have an older generation 1MP, that also is compatible. And I'm very excited to today inform you for the first time that we'll also have our SamuxMP, which is the mass photometer optimized for the use with AAVs. We'll have that in the SamuxMP, also coming with some new features available soon. So stay tuned. So to summarize, mass photometry rapidly and accurately measures the mass of label-free single molecules in solution. I hope that I could show you that we have a very quick sample analysis that takes less than 5 minutes from sample application to results. It has a very low sample consumption. It uses 10 to 20 microliters of sample at a concentration of 100 picomolar to 100 nanomolar. It has a very small footprint. The actual mass photometer is a small benchtop instrument, but also the robot that is obviously a bit bigger because it goes around it, but it's also still a benchtop instrument, so it doesn't take up a lot of space. We have single molecule resolution. That means that we can detect low abundance species and complex stoichiometries. And finally, it's a really easy experimental procedure with dedicated software and no sample preparation. Typically, it takes us less than 1 day to train a new user. With that, I'd like to conclude my presentation, and I would be very happy to take your questions. Thank you very much.